Good morning, afternoon, evening, depending on where you are. My name is Jack Dekkers, and I'm a co-PI on the Agriculture Genomes to Phenomes initiative funded by USDA NIFA. And this is our 19th field day already. And today we are going to focus on quantitative multiomics as a way to help unravel genotype to phenotype in crops and livestock. So we have two speakers, one on the livestock side, one on the crop side, to talk about some large-scale projects that they have ongoing in this area. And the purpose really is to discuss the infrastructure that is needed or available, the opportunities and bottlenecks in linking various omics data and to understand the genotype to phenotype mechanisms and networks. And we are first going to hear from Dr. Lingzhao Fang. Lingzhao is currently a postdoctoral researcher at the University of Edinburgh in the group of Albert Tunessa at the Institute of Genetics and Cancer at the University of Edinburgh. But in a month, he will be moving to Aarhus University in Denmark, where he will be a tenure-track assistant professor, and he will be leading a group that will continue much of this work that he'll talk about, focusing on both humans and animals, or animals including humans. And he's going to talk to us about farm GTEx, or farm animal genotype tissue expression. I'll turn the floor over to Lingzhao in a second, but if you, as usual, if you have questions, please put them in the chat and I'll monitor it and then interrupt the speaker if the question is immediately relevant to what he's talking about. But otherwise, we'll just address the questions at the end of the presentation. So with that, Lingzhao, I'll turn it over to you.
Okay, thank you very much, Jack. Thank you very much for the kind words. I think I may start to share my screen right now.
Yeah, go ahead. Nicole, on share. Yeah, okay, there we go.
Okay, so hopefully it works. So let me know.
Yes, yes.
Is that okay? Yeah. Okay, great. Thank you very much for the invitation. And this is, I think, is a really great opportunity and to introduce the community about the farm GTEx. And also, I will report some primary results from the farm GTEx in this presentation. So, about this project, the main aim about is to identify the genetic variants that are associated with the molecular phenotypes, especially for the gene expression across tissues in the farm animals. And so I will, okay, just before I started to the science, and I would like to thank all these colleagues from this institute, and because the farm GTEx is really an international consortium and try to put all the data together and uniformly analyze and then build publicly available resources and to allow us to understand how the genetic information flows from the genome to the phenome. So my topic will cover this as some backgrounds and the pilot phase of farm GTEx, but I will focus on the results from cattle GTEx and pig GTEx, but we're also doing chicken, sheep, and goat GTEx currently. And I will also talk a little bit about the next step for farm GTEx. So let's start with the background. I think most of us are quite familiar with GWAS, or population genetics genomics, that are trying to understand the genetic variants associated with complex phenotypes that we are interested in. And so here just to give three very interesting examples, like a height in humans is the well-studied complex traits. And right now, with over than 5 million individuals who can detect over than 10,000 independent loci that are associated with these complex phenotypes in the European population. And in dairy cattle, for the dairy traits like milk yield with 30,000 individuals, and the close to about 300 independent loci was detected, and also some about the adaptive evolution in the three bird populations were also reported several years ago. So we can see a lot of peaks that are related to the complex traits and also to the adaptive evolution. But we'll look into this loci, these peaks. Most of these these names are just the non-coding, located in the non-coding region. So it's super hard, also very challenging for us to really understand the underlying molecular mechanisms of these loci. So just to focus on the animal genetics and the livestock, also to chickens. And so you will see, look at the animal QTL database, we can see the QTL, the number of QTLs were detected, which increased a lot, according to the years. And right now, the thousands of these QTL regions were detected for hundreds of the complex traits of economic value in the livestock. So all this also is means that it's quite challenge. Also, it's really a high time for us to detect the causal variance in each of these loci and to pinpoint the molecular mechanisms. And we don't want to do this individually because it takes a lot of time. And how we can to identify the predicted causal variance in a system way or as scale. So that could be the way we can use integrative genomics by collecting the data. So here from GWAS population genetics, we know there are some associations become between the genetic variants and phenotypes we are interested in humans or in livestock or plants. But what I'm missing, there's a big gap over here, how this information flows through these intermediate layers of the biology, like how that influences the epigenetic changes or the gene expression changes in a particular tissue. So all these molecular phenotypes become possible in the current years due to the advances of the biotechnology. So we can collect these informations in different tissues, development stages, also different disease status. Because all these molecular phenotypes is quite context specific. Once we got this information, this can allow us to develop the more advanced statistical models and to truly understand the underlying circuitry, the regulatory circuitry of these complex traits. And then we can have the opportunity to manipulate this. And there are a lot of the big projects have been launched and to target on each of the layers. And today we got focus on the transcriptome level. Once we have this information, so it's much closer, and we can closer to the precision medicine in humans and probably precision breeding in livestock and plants. Start like I said, and this is a well-known project in humans and called GTAX. It's a 10 years project and but now there also have extensions like development of GTAX has been launched several years ago. But in this project, they collected the RNA-seq data in multiple major tissues, about 50 tissues in 1,000 individuals. And before they collected the gene expression data using RNA-seq and the whole genome sequence data and to detect the genetic variants. And then they associated the genetic variants to gene expression or alternative splicing and built a very powerful resources and to link these the resources of the regulatory variants across these tissues. And this also show very important to understand the genetics of complex traits in humans. So inspired by this very successful human project, we started to think about, can we do the similar work in farm animals? Because we we are facing the same challenge to detect the causal variance and understanding the causal, the molecular mechanisms. So the FARM-GTAS just trying to build a project or consortium and at a global level, and to collect this data and to analyze data uniformly and provide the reference, and like this intermediate phenotypes across multiple tissues, and just try to open this black box between the associations of genotypes and phenotypes. So we finished a cattle 2 years ago, and the paper, you can find the details if you are quite interested in the bioarchive paper. The paper will be online in Nature Genetics in the few, I think in a few weeks. And then right now we're also almost finished the peak, so we'll got the results and the paper in bioarchive in 2 months, and hopefully, and our chicken, sheep, and goat. So once we build these resources, it's really useful, just like showing, demonstrated in human GTAS. It will help a lot for understanding the genetic, the results from population genetics or genomics, and to really apply this information to the animal breeding area to increase the genomic prediction accuracy. And also, this also allows us to do the comparative genomics at a single base level, and also can guide us to develop the advanced biotechnology, like gene editing. So this is the global overview about the FARM-GTAS project. So that's including the pilot phase. And in this phase, we are trying to reanalyze all the public available RNA-seq and whole genome sequence data. And after this phase, and we will have the main part of the FARM-GTAS and try to cover more tissues and more development stages of sex, also genetic difference. And maybe the last, the final part, we'll try to move this from from gene expression and to other molecular phenotypes like epigenetics or protein. But we also move from bark tissue to cell types. At the end, we're just trying to build a very comprehensive regulatory elements, regulatory variance in the farm animals, and then try to apply this in different areas. And then understanding these key biological questions and in the system, but in the system genomics well. So this is the so right now I'm going to move to the pilot phase of the FARM-GTAS. I just trying to show some examples and to show the results we got by using the public data. So at this stage, the FARM-GTAS just focusing to add the values of the public data, because in the last more than 10 years, there are thousands of RNA-seq and whole genome sequence has been generated in each of these farm animal species, by the global community. And can we analyze this, use the uniform pipeline, and then clean this data and to get some useful information. This is very important. And then use this information to gather the next phase of the FARM-GTAS. So from analyzing all the public RNA-seq data, we can build the largest gene expression atlas, and to use this information to note the genomes in these farm animal species. And then we also can use the whole genome sequence data and to build a very powerful genotype imputation reference panel. And then by integrating these two resources together, and then we can detect the eQTLs in the major tissues in these farm animal species. And they integrated, then apply these resources and to interpret the GWAS results. And so we also can develop some like a TWAS database and to link the gene expression with complex phenotypes. And then let's quickly look at the major results from cattle GTAS. So I started at the beginning of the 2018, and then it takes more than 3 years and to finish this pilot phase. But we will start the next version very soon. Now, so the catalog GTAS just started and with the gene expression atlas, because at the beginning of the 2018, we analyzed the RNA-seq that are publicly available, only focused on Holstein breeds. And so after analyzing all this data, and we cluster the samples using the row gene expression, and we can see the tissues can be clustered. The samples were clustered quite well according to the tissue types, even though generated around the world in different labs. And so we can see that at least this public data can reflect the tissue biology. They show tissue specificity. And then we know the RNA-seq is kind of sequence data. Even though just focused on transcript region, but they're still, it's possible to call genotypes from the sequence data. And then at the end, we can see we can get about 50K SNPs from a normal RNA-seq. And then we start to ask, can we use this, use 1000 Whole Genome Project is the reference, and to impute this genotypes and to the searching level of to the certain sequence level. And at the end, we show that we can do a good job for the impute these genotypes to over 3 million SNPs. And then we'll believe that probably an in silico catalog GTAS is possible. So, and then we apply this pipeline to all the public available RNA-seq data at that time. So it's including about 10,000 RNA-seqs. By analyzing this uniformly, and then after the clustering of these samples, we can say these samples can be clustered quite well according to their tissue types. And even they come from different breeds or subspecies even. And then, so after the imputed the genotypes and based on RNA-seq SNPs, we can get 3 million SNPs for each of the samples. And then at the end, we got over 20 tissues, exactly 23 tissues where we have over 40 individuals. So we conducted the cis-eQTL and the cis-splicing QTL analysis in each of these tissues. As shown in panel A and panel B, you can see the sample size. So each of the dots means each of the tissue. And the sample size are linearly related to the eQTL power, as the y-axis shows the number of E genes divided by the number of tested genes. And if you look at the panel C, you still can see these tissues were ordered based on the sample size from smallest to largest. You will see you have a larger sample size, you got more power to detect the more independent signals, the eQTL signals per gene. And the red dots just means the mean number of the independent eQTLs per gene in this tissue. But all these results still show that additional data are really required to increase, especially here, because we normally no power to detect the eQTLs for some tissues. And also a lot of tissues are not covered in the public domain. And then we'll apply this information, these eQTLs and SQTLs, and to understand the GWAS of the complex trace. I just show two examples here. The left panel is kind of eQTL enrichment analysis. So here, this is for ketosis. We found that the eQTLs detected in liver was highly significantly enriched with the top associations. And for the milk yield, the TWAS also give a lot of the genes whose expression, for example, the DGAL1 expression in liver was the top association with the gene expression, with the milk yield, and also other genes and in their corresponding tissues was associated with milk yield. And then we also do some colocalization analysis to detect if there are any shared genetic variance between eQTLs and GWAS loci. So here just one example, this protein yield GWAS. And we can say that's perfectly colocalized with the eQTL for the DGAL1 in liver. It's a very nice correlation from here. Now let's look at the results from the PIG test. Also, it's very promising based on this public data. So in this PIG test, we analyze more data compared to cattle. So including over 10,000 RNA-seq and then over 1,600 whole genome sequence data to build the reference panel, and also the public available epigenetic data of single cell data that were used to do to functional note these eQTLs. And also we collected the GWAS, and all the GWAS were conducted based on the imputed sequence variance. That's including over 260 complex phenotypes. And we also have some validation populations to validate the eQTLs or the GWAS results. At the end, in this PIG test, and we got the eQTLs in 34 tissues where we have over 40 individuals. So the eQTL are conducted in each of the tissue and carefully corrected for the population genetics and unknown confounding factors. The B panel, the similar results. So the sample size is the key factor, associated and influencing the eQTL powers, and also very important for the fine mapping, to fine mapping of the eQTL signals. And then we also look at the tissue sharing patterns of these eQTLs. And so this panel, this here map just shows the cluster of the tissues based on the effect size of the eQTLs. So you will see these tissues were clustered quite well if they are from the same tissue system. They have the similar biological function. Like all the gut tissues are clustered here, and the brain, the yellow one, the different brain regions were clustered quite well, and also the immune, and like the immune tissues were clustered together. But it's very interesting to notice that the testes were quite different from other tissues because testes have a very distinct transcription profile, and this was observed in human GTACs as well. And besides the eQTLs, we also detected for protein coding genes, we also detect eQTLs for linker RNA genes, and also for individual exon and the enhancer regions. We also have the splicing eQTLs. But in general, you can say for the protein coding gene eQTLs, they're more likely to be tissue specific, or they could be shared across all the tissues being tested in the current data size. For other types that like individual exon or splicing or linker RNA, they show a more, they have a stronger tissue specific pattern. And then we apply these resources to understand or to interpret the GWAS loci, because we got the GWAS loci. So this is the total number. So over 1,500 independent GWAS loci from these 268 complex traits. So at the end, we show over 80% of these loci could be interpreted at least of one of these QTLs. And for each of these QTLs, splicing eQTLs, they have some specific, colocalization with the GWAS loci. So the B panel is just a very interesting example I would like to show you here. So from the TWAS analysis, and we found this ABCD4 gene whose expression in small intestine have the strongest association with the average back fat thickness. And if you focus on this ABCD4 gene and to check these TOR signals across all the tissues where we have information, and we'll also see a very strong association in brain regions. And if you look at the colocalization, so this Manhattan plot, this is from the GWAS, and this is from the EQTLs, and from brain with denoted as yellow, and the blue is small intestine. You will see the two signals have a perfect colocalization, and there are two SNPs were colocalized in these two tissues. And if you look at the chromatin state with the regulatory elements around these two SNPs, you only can see the enhancer regions in the gut tissues and in the brain regions, and no enhancer regions in other tissues. So it's a very interesting result. If you check this gene in humans, whose expression in brain and in gut tissues also associated with BMI, also weight. And I just want to show you some specific colocalization between EQTLs and the GWAS loci. So the left panel, this is the specific colocalization between the EQTL. EQTL just associated the genetic variance to the average gene expression. And this is the target trait, they call average delegain, the GWAS. And we only can see the colocalize for the EQTLs in colon, and this is a target gene, and no signals for splicing QTLs and the individual axon for the same gene. But to check another example, this is the specific colocalization between splicing QTLs and the GWAS loci. This is the target tissue called average bad fat thickness. And if you look at this gene, and you didn't see any colocalized signal, no signal in small intestine for the average gene expression, the EQTL, but you can see a perfect colocalization for the splicing, for the alternative splicing patterns of this gene in small intestine, but not for individual axon expression. And we also compared the genetics of the complex traits between pigs and humans. It's just based on the TWAS results, because this is the first time we got a large scale TWAS in pigs, another mammals in humans. And then we start to look at in the matched tissue, if there are any statistically significant correlations between the orthologous genes in terms of their effects with similar complex traits. So the A panel, just one example. This is the BMI, and this is the target tissue is brain. And BMI is in human, and in pigs, we got the days is quite similar to BMI. It's just indicated how many days it takes for the pig to grow to reach 100 kilograms. And then we can see the effect size across all the orthologous genes even have a weak correlation, but very significant. And we found some genes that are shared between these two species. And then we searched all these different kind of the GWAS in the UK Biobank, and we found a lot of these significant correlations. So this is just some primary results from the pilot phase of from GTAS, and you will got more details if you look at the preprint. And for the next step of from GTAS, and we are trying to, right now we just collaborate with FAN, and also new TF was launched just a month ago, and called the from GTAS TF, and trying to achieve some goals in the next futures. And the first is the aim is just trying to increase the sample size and including more tissue cells and more breeds. And this move the GTAs and to the contact specific GTAs, like to look at the developmental GTAs or sex interaction with the or genotypes or lactation and GTAs in dairy. And after that, and let's move to the enhanced farm GTAs, trying to including more molecular phenotypes, not just the gene expression. We also can collect the epigenetic information at a scale when the sequence caused the drop down. And we also can look at the microbiome and also protein level. After that, we also need to think about how to move this study from bulk tissue to single cell level. And this could be collaborated with another TF with Infang called single cell TF. So we know that for each of the bulk tissues, there are a mixture of cell types. And at the end, we need to detect the cell type specific QTLs and our cell state specific QTLs. Then we can, got more information, and we got more ways to interpret, to intervene this, and then will help us to develop more advanced biotechnology for the animal breeding. So at the end, I would like to summarize this. So the FarmGTAs is just an open collaborative project. The current stage trying to add the values to a public available data size. And right now we also have the TF and trying to encourage people around the world and to get involved in this project and to apply the relevant grants. And so this project, and we also publish in timely manner and release the results and make the results publicly available as soon as possible. We're also trying to develop the collaborative tools and to enhance the collaboration. So the reference panel for each of the farm animal species we plan to update it every 2 or 3 years. And at the end, I would like to thank all these colleagues, for all the colleagues from this institute and for their very strong support, and especially for George, Albert, Paul, Bingjie, Huaijun, and Meta, and to organize like the introduction workshop for FarmGTAs, also the current TF with Infang. Thank you very much, and this is all the results I would like to share with you today. Thanks.
Yeah. Okay, thank you very much, Ling Zhao. Very impressive work, and it's interesting to see what can be done by mining publicly available data. So if you have a question, please put them in the chat or just speak up. I'll start with one. So so this publicly available data is, most of these studies are pretty small, a limited number of animals, and they have typically a treatment comparing, right? And so looking at differentially expressed genes. So how do you, can you talk a little bit how you deal with this, the heterogeneity of this data?
Yeah, that's really challenging, because this I think we did a lot of work and try to gather metadata from the public published literature. But there's still a lot of metadata missing. But we analyzed all this data on ASIC data, use a uniform pipeline, and then try to cluster them together. We removed the samples were far from, for example, it was labeled as a liver, but it's far from other livers. So we just put a very stringent pipeline to remove the poor quality data. And the data we are not that was not with a good quality. And so we almost removed more than half of the RNA-seq data. So at the end, it's only half was returned. That's the big problem. That's the reason we need to have this plan, the project, the FarmGTA project, and so people can work together and to generate a more uniform data sets and to collect to get more metadata information. So for collecting, the batch effect in the public data, there are a lot of methods has been developed. This is very challenging as well in human. So that some methods can be used to remove that unknown batch effect and to keep the biological variance. And so we do try something at the end. We show the peer factor probably is good enough and at the current stage, but we also have some students are benchmarking these methods currently. Yeah.
Yeah. Okay. So there's a question in the chat for Mehdi Imam. In the best case of the association between a SNP and the expression of a gene, how much of the variation was explained by this SNP, and what was the coefficient of variation?
Okay. Yeah, that's a very nice question. We do check the heritability for this. Just to focus on cis heritability of the genes. The SNPs around just located 1 megabase around the TSS of the gene. So the average cis heritability is around 0.2. The trans heritability is a little bit higher, but we do not have enough sample size allow us to do the trans EQTL detection. We also have a current publication led by Yue Dongxiang, and who also you can check that preprint, and who use this information and integrated with GWAS in Australian population, and this shows that the cis-EQTLs can integrate it over 17% of the heritability of complex traits, so it's very promising. About the coefficient or variance of the genes, I do not have an idea about exactly the number. I will try to. I think we're gonna have a Slack channel, but I will try to answer over there. Yeah.
Okay. Yeah. Sounds good. Yeah, appreciate it. Question from Chris Tuggle. Follow up on the data heterogeneity. Are you using metadata standards? And that could be also useful for organizing the collection of new data from collaborators.
Yeah, I think that's it's a very, very nice suggestions. For the meta data standards, and right now, I think for the gene expression, there are a lot of, but right now the tissue, I think this is the key driver for the gene expression, the variance, and also the developmental stage. If the embryo or the adult tissue is quite different, and all others the treatment, I think. But right now we don't have a very standard, but we are using the public data set or trying to detect it, which is the key factor associated with gene variance across the individuals, and then probably later we can give a very standard meta, at least some information need to provide it based on their association with gene expression. Yeah, I think that's something we need to do in the coming.
Yeah, no, that would be very helpful. I have another question. So you made a comparison to the human GTEx, and of course, on human side there's a lot more funding available. But does the livestock data you have, I mean, is there an advantage? Could the human side learn something from what we're finding in livestock?
Yes, yeah, that's something we are working on, because we that for especially let's take a pig as example. We know the pig population were under very strong artificial selection. There are some functions of loss of function variance were selected at a certain level, and that genes were, because the human population is under natural selection. So some of the genes they have a relatively higher mass in the pig population. So you will see some of the genes that can be easily detected in the pig population associated with like weight and body size in pigs, but not detected in humans. If you check that gene will under very strong negative selection. So the mark was quite low. And I do think that the pig results can help a lot in humans, but in another way, the human results can also help a lot for pigs to detect the causal genes, because there are no LD in the two species.
Yeah. Well, of course, I mean, of course, in animals, in livestock, the tissue collection, can be a lot more designed, right?
Yes.
Compared to humans. I'm always surprised with the human data that, if you think about where those tissues come from, that they get meaningful results out of it, right? What are your thoughts on that?
Yeah, you mean the tissues from humans?
Yeah, that's right.
Yeah, I think there are a lot of limitations, in collecting tissues in humans, and so if you look at the metadata, they're quite different. There's also a lot of noise in the human, even in the well-designed studies. And I think in livestock we have a much better design, because we can raise all the animals within the same conditions, environmental conditions, and collect the data at the the same time point, and also at the same body size. This is I think this is a very important thing compared to the human studies, and this can later, I think these results with informed details can also help a lot in humans, because human people, for example, it's quite difficult for them to study the developmental, because it's so very hard to generate that data in uniform. But that's much easier in pigs, at least in pigs and chicken.
Yeah. Are there similar initiatives in lab animals like mice and?
I don't think I'm not pretty sure if mice can do this, because a lot of mice are inbred line. So you just need to randomly pick one from each inbred line and then sequence them. So the sample size cannot. I think they are not an autobred population. So the at least I didn't see any really related project in mice and also in fruit fly. Yeah.
Yeah, there is the what's it called, the collaborative cross in mice, right? Where I mean, similar to the NAMS population in plant breeding. So they.
Yeah. I think for mice, probably much easier, to look at a little specific. You can cross different inbred line and then generate the data and look at the little specific expression, and this also give you a lot of power to detect it. Yeah.
Any other questions from anybody? Speak up. If not, well, Lingzao, you'll stay on, and there may be some questions that come back to you at the end after Justin's talk, but thank you very much. So our next presenter is Dr. Justin Wally, is an associate professor in the Department of Plant Pathology and Microbiology here at Iowa State University. And he is going to talk to us about unraveling signaling networks using integrated omic profiling. So Justin, floor is yours. You're muted. Okay, I think.
Okay, how about now?
Yeah, we're good. Let me see your slide. Thanks.
Yep. Thank you. Thanks, Jack, for the introduction, and thanks for the ability to talk about some of the work we have going on in the lab. I'll focus on two main stories, one in Arabidopsis and one in corn, that kind of show and highlight how we're integrating these multi-omics data sets to get at phenotype. And what kind of drives the work we do is that is this idea that it's the proteotype that is driving phenotype. And by that, I mean, we know genotype. We know that development and environment constrain what's going on with the genotype. So the G by E form what we're calling proteotype, which is the proteomic state that uniquely underlies a phenotype. So again, what we're really trying to understand is the proteome or the proteotype in a lot of our work and our models. And one reason why we think the proteome is important is work I did when I was a postdoc with Steve Briggs at UC San Diego that I just wanted to highlight for a few slides here. And that was developing this developmental expression atlas for corn, where we looked at transcript abundance and protein abundance across a large range of developmental stages. And this data set, one provided a nice resource for the community, and the expression data is hosted at MazeGDB for this, for example. But it enabled, because it's such a large data set, to ask a few questions that hadn't been addressed before by studies that focused specifically on the transcriptome or specifically on the proteome. And one thing to highlight, and now this here has been shown by lots of groups and lots of eukaryotic organisms, it's not specific to corn or anything, but that this idea that there's a modest correlation between transcript and protein levels. So here's across 23 of the tissues where we have a Pearson correlation between transcript level and protein level of about 0.5. And again, that has been known for a while. But what we were able to show, because we had this integrated set of data, or we were able to integrate a set of data from a common set of tissue, is that topology of different types of networks are very different depending on the data that you use. What I'm showing here is a gene regulatory network. And the idea in these gene regulatory networks, which I'll talk about throughout my talk, is that you have transcription factors, and what you're modeling is the transcription factors abundance, and then what it's predicted to regulate. So what the regulated or predicted set is in all these examples is transcript abundance. And what we're changing between these two different gene regulatory networks is how we quantify transcription factor abundance. One network, we're using the transcription factors transcript abundance. Another, we're using its protein abundance. And then what we did to score how these networks performed is to look for conserved predictions between these two types of networks. And what we found is where there's about a 10% overlap in the predictions if you're using transcription factors, transcript abundance versus its protein abundance. So again, we're getting a very different topology depending on the data type. But one thing to highlight from this work is that when we integrated ways we've quantified transcription factors, these include, again, transcript abundance, protein abundance, but also phosphorylation state of the transcription factor into a unified framework. These resulted in the most accurate predictions. So using all these data types to form one integrated prediction was most accurate. And that's that ability and the kind of utility of integrating these data types is what's kind of driven the work in my lab. So the first story I wanted to highlight looks at brassinosteroids, which are a plant hormone. The signal transduction cascade is really well known, where you go from kinases at the membrane through a set of phosphorylation steps, ultimately regulating transcription through these master transcriptional regulators, BES1 and BZR1. So again, this initial signaling is really well understood, but how the global kind of response to brassinosteroids is modulated is an outstanding question. So that's what we sought to address. For this, again, we use transcript quantification, but also protein abundance and protein phosphorylation by mass spectrometry. For this, we grew Arabidopsis seedlings now on plates, and at 7 days old, we treated them with the hormone brisinolide for a series of time points, so ranging from 15 minutes all the way up to 8 hours. So we have a temporal response to brisinolide, and through this, we find lots of accumulation of gene products that are altered in response to brisinolide. And what I wanted to mainly highlight from this is you can see that transcripts responding to brisinolide, they're changing actually a bit later at points in time The protein abundances are changing kind of at an intermediate time point, and we get very rapid phosphorylation changes that are occurring in response to brisinolide, and they kind of peak in their DE a little bit earlier. So it's consistent with this signaling cascade driven by phosphorylation through changes in protein abundance, ultimately resulting in transcription. But this is a large set of data. We like to simplify it, and again, using these integrative network approaches. So what Natalie Clark did in the lab is develop this spatiotemporal clustering for integrative omics networks pipeline. It's available in GitHub. You can download and use. And the highlight of this is that it enables inputting different data types to get a single regulatory network framework. And this was a challenge going in because a lot of the inference pipelines that are out there are designed around specifically transcript profiling data sets. And the 4 key input features of this SCIEN pipeline are a list of regulators. These can really be anything you want to call a regulator. So if you're doing a gene regulatory network, you input transcription factor abundance. If you want to know what kinase is or in a kinase signaling network, you can input kinases into this as a regulator. You can change this to how you see fit. You have a list of potential target gene products. So again, a GRN, that would be transcript abundance data in a kinase signaling network. This potential target could be phosphorylation sites. We've also used this for metabolite data. So if you have enzymes as a regulator and all of the metabolites you measure, you can model that as well. And then you have your expression matrices. And if you want to do clustering, you can have a profile for clustering. So you input these different types. We have a few different preset clustering methods. I believe right now it's hierarchical clustering, dynamic time warping, and ICA. So this enables you to pre-cluster your data into modules, and then you can specifically run each module independently through a machine learning algorithm, Genie3, which actually creates a specific subnetwork in that case. So if you're making clusters in this step, you can do a gene regulatory network for each module or subcluster. And then that results in building one network for each cluster. In the second half of the talk, I'll talk a little bit about QTLs. You could also, instead of clustering the data, you could take all the genes in a QTL and use them as a potential regulator to infer what's the causal gene underlying a QTL. But ultimately, what you get is a merging of the subnetworks to form a final regulatory network. And another kind of network I want to highlight, or just highlight a bit more, is kinase signaling predictions. So I've mentioned gene regulatory networks again, where transcription factors are used to model the transcriptome or RNA abundance. And the kinase signaling network, what we're doing is taking activation state of the kinases that we measure with proteomics and using that to model what potential phosphorylation sites they're regulating the phosphorylation state of. And how we're getting this activation or kinase activation state information stems from eukaryotic kinases have an activation loop. And when this activation loop is phosphorylated, it changes conformation of the kinase into its active form. So what we're using is phosphorylation of a kinase in its activation loop to predict where and when the kinase is actually active, not where it's just simply present. And what we ended up with in this brasinosteroid context is a merged network where we merged the gene regulatory network and the kinase signaling network. And we're kind of highlighting the different data types in different colors. So abundance would be abundance of the transcription factor, making the gene regulatory network predictions. Phosphosite would be phosphorylation state of the transcription factor and how that's modeling or in those predictions. And then the purple would be the kinase signaling, so the kinases and their phosphorylation sites. And the way this little network is overlaid here, it's lined up so that time is at the top. It would be the early time points, and time at the bottom would be the later time points in our brasinosteroid time course. And you can see that it's modeling with temporal specificity that kinase signaling events are predicted to be happening earlier. Transcription factor state of the phosphorylation state is important at an intermediate time. And then these changes in transcription factor abundance are making the later time point predictions. So we've recapitulated kind of what's known about the timing of a Brasenose steroid response. And then to highlight the utility of making these predictions, I have two things I want to show. And then when I showed the initial Brasenose steroid response, two of the key factors in that well-characterized cascade are this kinase BIN2 and this transcription factor BES1. And it's known that BIN2 phosphorylates BES1, but despite decades of study on these two proteins, most of the actual sites on BES1 that are phosphorylated were unknown. And what we were able to predict with this network approach is two specific sites on BES1 that are phosphorylated by BIN2, and the prediction is that they're important for BES1's function. So we then directly studied these two phosphorylation sites. The first way is an in vitro kinase assay where we mutated those two specific serines to alanine, making a phosphorylation null form of BES1. So it's present, but it can't be phosphorylated. And what we found in vitro when we had recombinant BIN2 is this phosphonull form of BES1 is phosphorylated to a lesser extent, consistent with these being BIN2 phosphorylation sites. And then when we made transgenic lines expressing this phosphonull form of BES1, we end up with Brasenose steroid gain of function phenotypes consistent with BIN2 being a negative regulator of BES1. So again, we've used this network approach to find new sites that are important for BES1 function on these two well-characterized proteins. And then another more global way to get at things that may be of interest in these large networks is to use network motif scoring. So here, what you're doing is scoring different types of motifs in a network. So feedback, feed forward, diamond motifs, etc. So these are motifs within a network that are thought to be biologically important. And you score simply how many times a gene of interest is present in one of these network motifs. And what that led us to is this transcription factor, which hadn't been studied, which we ultimately ended up naming Brontosaurus. So Brontosaurus is shown here. Upstream of Brontosaurus are a number of well-characterized kinases that are predicted to phosphorylate an upstream transcription factor. And when that transcription factor is phosphorylated, it is predicted to regulate Brontosaurus. Also important is that Brontosaurus is in a feedback loop with another VDOF transcription factor. So that's the framework of our predictions from this network reconstruction. When we look specifically at loss of function mutants of Brontosaurus, these are tDNA insertion mutants we obtained, we found that they're hypersensitive to prasinolide treatment. So here is root length growth assays where you have it plus minus prasinolide, and you can see that these mutants are hypersensitive relative to the wild type control. And when we image the roots, we find that these loss of function mutants have more meristematic cells and divisions in the quiescent center, showing how they might promote hypersensitivity. And then what we wanted to do is kind of validate, the specific aspects of our predictions. And I'm not going to show the data, but we used a combination of quantitative PCR on loss of function mutants of these events upstream of Brontosaurus, as well as RNA sequencing of the Brontosaurus mutant. So these kinases, which were all predicted to be upstream of Brontosaurus, we again got loss of function mutants of them, and then looked at Brontosaurus transcript levels, and we were able to assign whether they positively or negatively regulate Brontosaurus transcription. And that feedback loop I mentioned with the VDOF transcription factor, we recapitulate that where VDOF is a positive regulator of Brontosaurus, whereas Brontosaurus is a negative regulator of VDOF. So we've, kind of confirmed those upstream predictions. And also coming back to the cell division phenotype, Through the RNA sequencing, we found a number of cyclin-dependent protein kinases, which are known to be involved in BR-regulated root growth, are misexpressed in Brontosaurus. So we believe that's how the cell division is altered. So this is one example of how we can use multi-omics data to integrate into predictive networks and then kind of get down to the nitty-gritty phenotype through different ways to simplify the data. And the second project I want to talk about is a project in corn, where we use system genetics to look at northern corn leaf blight resistance. This was led by Natalie Clark in my lab, who was a postdoc and is now a staff scientist at the Broad Institute. And Guoyan Song is still in the lab. And it was a really good collaboration with Nick Lauder's group, which made a lot of this possible. So in this, we used a intermated B73 by Mo17 recombinant inbred line population that Nick's lab had access to. They've done a number of years of scoring disease QTL within this population, and I wanted to highlight, that the two inbred parent lines are kind of intermediate in their resistance now. So this is disease scores to northern leaf blight. So this is the population we used. It's a larger population than what we used, but we ended up at 134 recombinant inbred lines grown in the field. We inoculated them with northern leaf blight, which totaled about 30,000 inoculations. Nick's group performed disease severity ratings, and then at 1 week, we collected tissue post-infection for the molecular assays that my lab has worked on. So what we were really interested in is, could we map different types of molecular QTL? We heard in the very nice last talk on transcript QTL or eQTL mapping. There's been some large scale reports of protein QTL, mainly in mouse in the literature. And what we were really interested in is, could we map phosphorylation QTL? So regions of the genome that regulate phosphorylation levels of specific phosphosites. So in this case, the phosphorylation site is the trait, and the QTL is regions that regulate that phosphorylation. And there haven't been any large scale reports in the literature on this. And I don't have this on here, but we also did metabolite QTL in collaboration with Sean Christensen's group. So we can find out, what specific set of metabolites are present and how they're regulated. But what this resulted in is quantification of about 30,000 RNA, 11,000 proteins, and 42,000 phosphorylation sites. From that, we were able to again map different types of QTL. So we have a large number of transcript QTL, 34,000, about 5,000 protein QTL, and about 5,000 phosphorylation QTL. And you can see there's a modest overlap, for example, between transcript QTL and protein QTL. And if you tease that apart into cis, so local QTL versus trans QTL, we can see most of the overlap is actually coming in cis. So these cis QTL account for a large percentage of the overlap we're seeing between the different data types. And then to kind of highlight a couple complex relationships between transcript and protein QTL, I'll give a couple examples. One is for this brown ribib 3 gene, and we find that it's either transcript or its protein abundance has QTL that regulate their specific gene type. And we find that they're non-overlapping cis QTL, eQTL, and pQTL. So they both have QTL at this, it has a QTL at an eQTL level and pQTL level, but they're different regions of the genome. And consistently, we find if we look across the different inbred lines and just compare BM3 transcript versus protein abundance, limited correlation. What we're really kind of surprised to see is another example for this gene PX9. Here we have overlapping cis eQTL and pQTL, both on chromosome 1. So the same region of the genome is regulating PX9 transcript and PX9 protein. But when we look at the specific data, we can see that there's actually a negative correlation between transcript abundance and protein abundance for BX9, highlighting that even, there's a shared QTL, you can have a complex interaction between their gene products. And then to kind of step back at a more global level of just, what kind of QTL are we mapping? In this case, we looked at synthetic genes with sorghum. So these are genes that are conserved between corn and sorghum. And we can see that we're more likely, or we do observe more synthetic genes as QTL traits. So these are genes that have QTL that regulate their either transcript protein or phosphorylation state. And if they're synthetic with sorghum, we're more likely to be able to map them. So suggesting that they're under stronger genetic control. Along that line, we downloaded publicly available protein-protein interaction data. So these are physical interactions now between proteins that has been generated by other labs, some of it experimental, some of it predicted. But what we looked at are hubs in these protein-protein interaction networks. It's been shown in a number of different studies that protein-protein interaction hubs, so proteins that interact with lots of other proteins, are more likely to be biologically important. And what we can see here is at the eQTL and pQTL level, protein-protein interaction network hubs are more likely to be traits of either a QTL, eQTL, or pQTL. So again, suggesting that these biologically important hubs are genetically controlled in a way that we can map. We did not observe an enrichment in phosphorylation QTL, and we don't know if this is a true biological phenomena or just simply because there was a very limited number of phos-QTL traits that were in these protein-protein interaction networks. So it was where we had 2,000 or so gene products to look at eQTL and pQTL. There was only about 100 phos-QTL traits in this network. So it could just be a limited sample size artifact. So, one of the ideas behind this work is, can we get kinases within a QTL that are responsible for the trait? Can we directly map kinases responsible for the phosphorylation, not just have QTL? So can we find and identify kinases under the QTL that are causative? We looked at this several ways, and just kind of a global picture, we find 56 phos-QTL hotspots. So these are regions of the genome that regulate lots of phosphorylation sites as traits. And actually a majority of these hotspots occur on chromosome 1, so over about 42% of them occur on that. And we looked kind of more in depth, I'll show in the next slide, at 3 of these hotspots which have a kinase in them of interest. One is this hotspot which has a kinase BAC1, this PKSP kinase, and LTK1 kinase. So we looked at these specific kinases and used an in vitro kinase assay. And what we are specifically using is this multiplexed assay for kinase specificity. So instead of doing a kinase assay in vitro where you have one substrate protein and one kinase, what we do is express the kinases, in this case, LTK1, PKSP, and BAC1 in E. coli, purify each of those kinases independently, and then incubate them with global protein extracts that were from the inbred parent lines B73 and MO17. So we have a global protein extract that we're incubating with a specific kinase. We perform our typical proteomics experiment now following incubation, where we have a Gus or Planck control, and then samples incubated with each of the 3 different kinases, perform phosphoproteome profiling, and look for sites that are enriched in the specific kinase, meaning they're phosphorylated specifically when that kinase is present. And what we can find for each of these kinases is that in these assays, the substrates in the MAX assay have a significant overlap with each of their specific phos-QTL traits. Supports the idea that our phos-QTL mapping enabled identification of kinases that are the causative kinase in that QTL interval. And then to get at what are these signaling cascades, and ultimately how is this impacting NLB resistance? We used the SciION pipeline I talked about early to infer causal genes underlying the different types of QTL. So in this case, each QTL interval, we made a specific sub network reconstruction. And then we merged all of these sub-network reconstructions into this specific large-scale network. So this will capture, regulatory events that are specific to NLB, but also just what gene regulation is occurring in the maize leaf at that point in time. So it can be useful not just for NLB studies, but just what is a signaling cascade, what's regulating what in the leaf. And there's a lot of information here. To simplify it into what we're specifically interested in, we went back to this kinase BAC1. So again, BAC1 had a large number of max direct targets. And additionally, BAC1 was of interest because a previous GWAS paper or publication had shown that NLB resistance is associated with BAC1. So now we've pulled here just the first neighbor network of BAC1 and then highlighting the edge color where the information came from, whether it's transcript protein or phosphorylation QTL. That was of interest. So this still had a large number of genes involved. I believe it's 200 and some. And we focused further on a specific subnetwork within this BAC1 first neighbor, which was BAC1's phosphorylation of this transcription factor BIM2 and its predicted regulation of the gene brown rib 2. So why we focused on this is that the MAX assay showed that BAC1 was also, in addition to it being, associated quantitative genetic sense of a PhosQTL, it's also a direct substate, which we can verify with the MAX assay. So BAC1 directly phosphorylates BIM2. And then why we were interested in this is that BIM2's regulation of brown rib 2. And earlier this year, Judy Coleman and Rebecca Nelson published that brown midrib 2 was required for NLB resistance. So now we have what we're predicting is an upstream cascade regulating a gene where we know is involved in NLB resistance. So we specifically tested if we ordered transposon insertional mutants in either BAC1 or BIM2, how are they responding to NLB? And what we can see here, this would be the wild type response to NLB infection. So this would be the percent of plants with lesions by day, and then also the incytosoina area under the curve, so the lesion area. And in both ways to quantify, we can see that these two BAC1 and BIM2 mutant lines exhibit increased susceptibility to NLB. So again, showing that we can go from these complex networks and find information we can verify as to the biological consequence of these specific genes. So in summary, we're able to map genomic regions that control transcript protein and phosphorylation levels. There's a moderate overlap between transcript and protein and QTL, and most of that arises from the cis QTL rather than trans. And we're able to make regulatory networks that enable identification of genes specifically modulating NLB resistance. So with that, I'd like to thank members of the lab and for the NLB, particularly Dr. Song and Dr. Clark. And again, the Brontosaurus work was also done by Dr. Clark. The NLB was made possible by a collaboration with Nick Lauder's group. Mercy was a grad student with him who did a lot of the QTL mapping along with Miriam. Sean Christensen did some of the metabolite measurements that I kind of glossed over. And then I left over out of here are on the steroid side. A lot of this was collaboration with Yan-Hai Yin's group here at Iowa State. And in particular, I want to highlight funding from the Plant Sciences Institute, which made the work possible to do the NLB systems genetics. And with that, I'm happy to take any questions.
Hey, thank you very much, Justin. Very interesting work, very insightful. I'll again put your questions in the chat or just speak up if you're on for those online. I'll start with a question. So if you had to prioritize the different types of omics data, if you wanted to get at the causative genes behind, yield in or resistance to a particular disease in corn, for example, how would you prioritize the different omics data that can be collected?
I would prioritize. I know from our work in Arabidopsis and corn and a couple other groups we've collaborated with, just looking at if we do an omics study and look at loss of mutant and then a follow-up mutational study, we have much higher success rates at finding genes with altered phenotypes if we're using protein abundance or protein phosphorylation data than transcript data. So transcript data would tend to be maybe 20 to 30% of the reverse genetics have altered phenotypes. The proteomics data were typically over 50% of the candidates we look at. So that's one way to look at it. I haven't done the cost benefit analysis. Transcriptomics is obviously a lot cheaper. And, I have a mass spectrometer in the lab, so we can do a lot of experiments at a scope that other groups can't. So that is also a constraint. So, but if I had the data available, I'd look at some type of protein data.
Yeah. Okay. Interesting. Another question, and maybe I can bring Ling Zhao back into this also, because both of you, you're trying to understand the genotype to phenotype, but using integrated omics approaches. But your approaches are quite, to some degree, different, right? And so, and there may be good reasons for that. So, Justin, in your case, you're looking at specific signaling networks and you're designing specific experiments, whereas Ling Zhao uses public data from very heterogeneous data. So maybe question to you, Justin, is there a crop GTEx? And if not, is there a need for a crop GTEx?
Yeah, I'm not sure, and I think it would be useful. I know there's a ZenCode effort to kind of look at modeling after the ENCODE in mammals to look at chromatin states, and that also includes expression data, but I'm not sure exactly what data types are going into it. Probably Pat Schnabel would have his finger on the pulse of what's going on in corn a lot better than me.
Yeah. Does anybody else online have information on, is there a crop GTEx? Or that I'm assuming there's a lot of public data there also, right? Yeah.
Yeah, I think there could be, opportunity to try, from some plants species, yeah.
But then on the other hand, in plants, you can, like you do, Justin, you can design your experiment and, large enough to stand on its own too, right?
Yeah. I think that I'm aware of a lot more of the large scale, kind of focused net aspects where like Ed Buckler's group has done a ton of expression profiling and chromatin state profiling across inbred lines and recombinant inbred lines and accessions. So
yeah.
But I'm not sure about the kind of multi-lab reuse of data.
Yeah. So Ling Zhao, to get to the kind of detail in terms of networks that Justin was talking about, what do we need in animals, or what are we lacking in animals to get to that level of detail?
Yeah, I think right now, for that network, I don't think there are a lot of the data, at least to my knowledge. I don't think any group generated in other scale generated a very like Justin generated a multi-omics data in the same animals. That's definitely a very interesting thing. And if we can't generate the data from the same individuals, and then we need to know for each of their data, we need the population level. That's a huge amount of work. Yeah.
Yeah.
to link the variants and to the to the each of these phenotypes. So I think that's also something in animals we can to think about how to generate, at least to build some tissue banks, and maybe for now we can generate the multiple layers or the omics data, but maybe once later we got ground, so we can continue to generate the same data from similar animals. That will be very interesting.
Yeah, and of course
but it's a little bit difficult, to store the animal tissues compared to plants, I think, but I don't know. Yeah, what do you think, Jack?
Well, in terms of collecting all types of omics data on the same animals, I mean, Fang and Chris Tuggle is on here. Maybe Chris can comment also. Fang is doing that, but of course on a very limited number of individuals, right? And I think we just have to expand that to a much larger number of individuals, but that requires a lot of funding.
I know storage wise, we have some work going with Josh Selsby here at Iowa State from Animal Science, where we're looking at proteomics in pig and mouse, and the animal tissues are much more protein dense. So, a 2 mil tube can store all the protein you'd ever want to use, whereas a lot of the leaf samples we're storing are in 50 mil conical tubes. So just in a freezer storage aspect of it, the animals can be a little bit easier to store.
But is it also correct for me to say that, I think one of the things that Justin allows the plant, on the crop side, the plant side, to get into a lot of these details, because there's a lot more known about the biology, the pathways. And I think we're lacking that in animals also, in the specific pathways involved. And, the systems are more complex also. But, Any questions? I don't see anything in the chat. Anyone, just speak up if you have a question for either Justin or Ling Zhao.
Jack, this is Chris Tuggle. I would add to what you said, and thanks for pointing out that Fang has been working on the first sort of stage with a limited number of animals, but clearly that's the next phase of Fang, is to move toward a limited epigenetics and functional data on much larger populations of animals. And that's sort of the overlap with what Ling Zhao said, in terms of, at least on the gene expression side. But there are people who are interested in molecular phenotyping at a number of levels, including phenotyping organoids and other animal models of animals. So that it's a very large open area. I think the question that you raised about what is the most information dense type of data for identifying genetic control, I think is a really good question. And that's something we're discussing as well. What's the minimum amount of epigenetics data to collect to characterize variation in function within the genome? And so it's a really important question that I don't think is resolved very well.
Have they looked at that on the human side? Anybody know of?
Yeah, some work has been done. I read some literatures and talking about which kind of data sets can give you more, like maybe based on colocalization analysis. So you will say if the layer of the data more close to genome, and that's information more powerful too. Like methylation QTLs probably can give you more information compared to gene expression and compared to protein, but I'm not pretty sure. That might depend on the complex traits. I don't see a very large scale data has been done just based on particular complex traits.
Yeah.
Yeah, that's definitely different across trees because the genetic architecture of different trees could be quite different. Yeah.
Mhm.
Yeah. That's a really good comments and suggestions from Chris Targo and how to generate this the useful information now from which tissue, which time point, and this is more important to related to the phenotypes and we are interested in. And yeah, that's definitely something we need to think about. Maybe the cell type single cell information data can give more compared to the bulk tissue, a mixture of the information. Yeah, at least in human, they reported a lot of information, but just look at the limited overlaps between the EQTLs from bulk tissue, like from GTAS with the GWAS loci. Yeah, so it's still a very open area to talk about this.
Yeah. Another question. So, AG2PI is very focused on overlap between crops and livestock. So is there, in terms of data and analysis pipelines, Justin, is there a reason why the pipelines that you have developed, given the right data available in it, could be used for animals?
Yeah, there's no reason to not use it.
Mhm.
And actually, Natalie Clark, the postdoc, gave this talk at the American Society of Mass Spectrometry last year, and that's how she got recruited to Broad Institute. So they're using it now in mouse and human studies.
Mhm. Okay, interesting. And Ling Zhao, from your side, is there a reason why your methodology could not be used to do a crop GTEx?
Yeah, I don't think any problem that can be easily attend to the crop. And just check the quality of the public data by uniformly analyze it, because most of these the variance is related to the bioinformatics pipeline you applied. And so if you analyze the data use a uniform pipeline, usually the technical bias will not that big. Yeah, but if you just download the process data because they're generated in different pipelines, and you will see a huge amount difference. Yeah. Just try to use the same pipeline to analyze all the data, and then use different methods and try to correct this noise, correct for the noise, and you will find some biological variance in this data. It's just there's a huge amount of work you need to try.
Yeah, that's right. Yeah. Okay, interesting. Any other last questions? Anybody? Just speak up.
Jack, there's one in the chat.
Oh, yeah, from Serge at me. With these types of studies and designs, are you hitting major genes or minor genes? And are you able to make understand the differences between those?
I'll use the Brontosaurus example from Arabidopsis. So I think we're getting minor genes as well as major. I'm not sure we're able to tell the difference per se, but response to BL is one of the classic screens that have been done in forward genetic screens in Arabidopsis. So they've been done repeatedly and exhaustively in a number of different labs. And we still were able to find a new gene with a minor, what would be called a minor phenotype that was missed in forward genetic screens.
Okay, good.
Yeah, I just got another question for Justin. Probably I missed that. Did you check if, because you look at the co-expression and like a correlation network of the genes, did you find the genetic variance, was associated with the core genes? The genes were located in the central part of the network. Are they different with genes are far from the central part? Because I think that genes located in the central of the network maybe are more important and they are more tolerated with genetic variance or not. That's something we are doing in animals. Yeah.
Yeah, we haven't looked at that. It'd be interesting to look at that.
Okay. Yeah, I think that's a probably because the genes they may under different, selection and constraints if they play different roles in the network.
Yeah, and going forward, these were recombinant inbred population with kind of large QTL regions where we want to do this in an association panel or a diversity panel so we can get more at gene level differences and get better at the causative gene. I think that would be better for addressing your specific question. But
Yes, yeah, okay, thanks. Yeah.
Good. Well, we're out of time. Thanks, Justin and Ling Zhao. Excellent presentations, and I think we had some good maybe opportunities for some collaborations across crops and animals. So for well, there's also Slack channel. So if you want to continue some of the conversation, please use the Slack channel. As you disconnect, there will be a survey, which we hope you'll be able to fill in and give us some feedback on this
Mm-hmm.
field day. So normally the field days are every third Wednesday of the month, but we're going to skip July. Our field day in August, we're hoping to, and this is an invitation to industry to present on some technologies or pipelines that they have, that they, as an industry collaborator, you believe might be of use for genotype to phenotype research in crops or animals. So if you're interested in that, on the website, there's a way to apply for that, fairly short application. So that's hopefully scheduled for, if we get applications, scheduled for the August field day, and then the September field day, we also focusing on industry, but more focusing on collaborations of industry partners with primarily academia. So collaborations that have worked well, if you want to highlight those. So if you're not in industry, but you are collaborating with industry, talk to your industry partners and suggest that they apply to that to highlight their collaborations and the opportunities and the bottlenecks. And again, the AG2PI website has the application, and it's also in the newsletter that came out, I believe, last week. So that's as far as the field days. There is going to be another workshop, a crop modeling workshop, June 23. And please sign up if you're interested. They fill up fairly quickly. And then in July, there will be one on a UAV pipeline for crop breeding, a workshop on that topic. We still have some seed grants available. There are seed grants available on for rolling or community event grants. If you want to organize an event that or a workshop that is related to AG2PI, please look at that. And then there's also working group grants. So if you want to form an interdisciplinary group to address or investigate or discuss opportunities for AG2PI research that is interdisciplinary, please look at that. And you can submit those applications anytime, and we'll fund those as funds are available. So with that, finally want to thank Nicole Scott also for the help with organizing these field days. And with that, I'll close the field day and wish you all good rest of the day and of the week. And thanks again for everybody's participation.